Chris, thank you for the introduction. And good evening, everyone. As Chris said, I'm very happy to be here because it's just only a couple of minutes from my office in the former Michael Faraday's apartments upstairs. But of course, not only because of this, but mainly because this stage hosted a lot of great scientists during the last 200 years to talk about science. And science is my passion for all my life, so I'm so happy and pleased to to tell you about science, what I'm doing. And I moved to London about 3 years ago, and thanks to Professor Carl Friston, who is here with us today, I joined London Institute for Mathematical Sciences and doing research there on making machines smarter. And today, together, we will try to chart a roadmap to the future of biology with AI. So let's start with some personal story. As I've said, my passion for science is defined by 3 simple questions. The first one is, what are the most fundamental principles of learning we have in nature or in machine? So if we know these principles, can we implement them in machine so it will learn like an organism? And the last one, can we use these machines to speed up, to accelerate scientific discovery? So we will close the loop and we will get better understanding of nature around us. And 10 years ago, I thought, and probably I was, right on that track. So you can see here on the right-hand side, these are real biological neural networks. So it's like neurons grown in the Petri dish with the macro electrodes embedded in the bottom of this dish. So what we can do with this, we can do experiments and study the most primary principles, how neurons learn with each other to control external stimulation. And in parallel, to devise theories and do computational simulation of these neuronal cultures, neuronal networks in the dish. But then I have read a paper in Nature, and this is not an ordinary paper. It's landed on the cover of Nature. And this is a paper about very new approach to train neural networks, artificial neural networks at that moment, proposed by DeepMind. And I was struck by this paper because before I've seen it, I thought, yeah, neural networks, they are very powerful, but for every specific problem, you should devise how to represent your problem for your neural network, and then you should train this network to solve your problem. What DeepMind showed, that you can take exactly the same architecture, which just observes the world through pixels, through screen of Atari games, through frames of Atari games. And architecture is the same, but games are totally different. So they have different rules, they have different laws inside the games, but you can train the same architecture with the same algorithm to solve many of these games better than humans. And this was astonishing and changed my world. So I decided that I need to do something in this direction. And but probably games, they are interesting, but they are very limited. So it's just toy worlds, and probably we will not be able to solve real-world problems by playing games. So I decided to think, what is the next big thing? What should we do to have a maximum impact from my research? And I decided that the next big thing is conversational AI and natural language processing. Because language is so powerful. You can describe anything about our world with language, and then do calculations, do simulations, do reasoning with language. So probably it's even more universal than universal game playing of Atari games, because here you have image and you have a joystick, and in language you have language as an input, language as an output. It's very universal and computer friendly. And you have a lot of data, a whole internet for you. So I've switched to conversational AI and natural language processing. Probably you already have a question, how all this conversational stuff relates to the topic of our lecture today. But hang on, you will know soon. Oh yeah, and of course, during these 10 years, when I've done this research, I focused on improving capabilities of these neural networks. So one of the most, one of the areas which interested me most is how we can provide these networks with some kind of working memory, like a scratch pad to think. So we devised a series of memory-augmented neural networks, and when we started to test them, we found that they are pretty good at processing long contexts. So we break a couple of historical records of the context length in neural networks. But let's get back to the main topic of our lecture. So why we are speaking about biology and AI? Because actually, life as a subject, as a phenomenon, is very hard to understand. Among all natural sciences, biology has like a less mathematical theory than any other natural science. Why? Probably because biological phenomena is generated by billions of interacting parts, and it's very hard to devise laws of these interactions. Of course, let's just take a short overview of our human body. We have 11 organ systems, and these 11 organ systems comprised by 78 human organs, and organs comprised by cells. So we have about 30 trillion cells in our body, and it's more than 100 types of cells. And every cell, it contains of more than 40 million proteins. And we have more than 10,000 different types of proteins in our cells. But what is more exciting is that all this complexity, it's encoded in genome, which for human consists of about 3 billion nucleotides or base pairs. And so you see, we have a lot of levels of organizations which interact with each other, and a lot of entities on each of these levels. So, is this problem solvable? Can we do something beyond what we are doing today in our science to understand biology? But we have example. We have example of another complex phenomenon, complex subject. It's our human language. You see, actually, every day we use language, and probably we are not thinking about how complex is it. But just imagine, when I'm speaking, I am generating sequences of acoustic waves. These acoustic waves reach your eardrums, and then they generate some impulses in your brain, so you can start smiling, or crying, or laughing. Isn't it amazing? And it's unique human capability. No other species has such developed language and use it for reasoning as human beings. So certainly human language is an extremely complex phenomenon. And to understand it, we need to understand common sense, how brain models like its environment, its surrounding world. So let's take a short overview of what we've got in language during the last 10 years. So in 2015, 10 years ago, new approach to study language started to emerge. This approach is called language modeling. So of course, even before that, like many years ago, we have some language models. But in 2015, we started to see new language models powered by neural networks. So let me give you just short overview of what language model is. And of course, it's some model of language, and it's pretty simple, because it is a model, here it's neural network model, which takes a character of your language. So we split long text on characters, and then we devise a model, we train a model to predict text character by character. So the model takes one letter as an input and predicts the next letter. So here it takes letter T and predicts letter E. And then we take this letter E and feed as an input to our neural network, to our language model, and ask it to predict next letter. Here it's letter N. And then we take this letter and feed it as an input, predict next letter, and so on and so forth. You see, like, training objective for this algorithm is so beautifully simple. So you need only to predict the next element in the sequence, next letter in your text. So what complex can emerge from this? I don't know. You see, you have a neural network. You randomly initialize weights. Probably in 2015 you have 100,000 weights or 500,000 weights, no more than that. And so you have some random language model, then you start to train it. And you introduce no vocabulary, so no vocabulary placed in the model. No grammar rules, no syntax, no semantics. All you have just texts, and you learn to predict next letter. Yeah, I was very skeptical that this thing can learn something 10 years ago. And when I saw some results presenting that actually these models, they can learn some coherent language, I was surprised. And I decided that I need to test it by myself. So we also took open subtitles dataset, which is just subtitles for TV series and movies. And then we trained like a pretty small network, very small network by today's standard for 2 weeks. And then I had chat. So this is something from the middle of this chart. So I'm asking the model, funny, but you are just a program and you will never be able to think. And the answer was, of course. The thing is, now is not the best time. You see that the model learned just from predicting next letter, all the capitalization, all the punctuation marks, all the English vocabulary here. I expecting that I will get like 3 words and then ... because before that I had pretty good experience with all these recurrent neural networks. So usually they cycle in some like short sequences repeating or repeating one element. So I was just amazed. Yeah, and that was 10 years ago. But now, you know, we take the technology developed over 10 years, and now we have all these beautiful, now they call it large language models. Why large? Because during these 10 years, we discovered that we have so-called neural scaling laws. These neural scaling laws tell us that the more data we train on, the more parameters our model has, and more compute we throw in to train this model, the better the results. So, but you can ask me, what is the principal difference between character level model 10 years ago and current large language models? Of course, current language models, large language models, they have a bit different architecture, but they fundamentally solve the same task. These new language models, they also train just to predict the next element in the text. Today, it's a word, not a letter, but still, objective for training is the same, and it's amazing. So you see, just by scaling architecture, you get better and better and better results. And these models, they can master some aspects of language even better than average human. So here is a party trick. If you take a ChatGPT and ask, like, give me a rap verse for something. Here is for first Newton's law. It can give you a verse in just, I don't know, 2 seconds, 1 second. But you can do even more. What you can do now, you can take this verse, take these lyrics, put it in another AI, and then wait for 1 minute to have a full track.
Yeah, yeah. Yeah, yeah, yeah.
Enjoy.
Yeah, I'm chilling in motion or I'm still at rest. A force gotta hit me, put my skills to the test. If nothing's pushing or pulling, I'll just keep my pace. Keep rolling steady or staying in place. It's inertia, baby, that's the name of the game. You feeling that resistance? Yeah, Newton's to blame. To blame, to blame, to blame.
So now you know whom to blame for inertia. Okay, so probably now it's clear where I'm heading to. I want to say that if large language models helped us to understand such a complex phenomena as a natural language, can we use the same approach to understand language of life, language of biology? And this is what I will talk about today. But before we start with AI applications in biology, let's do a small recap what artificial intelligence, or AI, is. So here I will contrast it with normal programming. So if you want to automate solution of some task, first, of course, you need to know in step-by-step recipe how to solve this task. So you need to know solution beforehand, before you start programming. But machine learning, or AI, it just turns it upside down. It says that, oh no, it's so hard to solve the problem, I don't want to do this, but I know if something is wrong or not wrong. And then I'm so smart that I can devise algorithm which will train another program to solve the task. And this is how AI learns. So you have some annotated data, some training data set. In computer vision, like in image classification task, it might be if you want to differentiate cats from dogs, you will have images from cats and dogs, and then you annotate them, and then you provide your neural network with these images, and your network starts learning by making errors, and you use these errors to change parameters of your network to decrease this error. And after that, you find solution which works much better than solution you propose for this problem and program. And like basic block for the current AI systems are neural networks. So let's take a look at biological brains to have idea how artificial neural networks work. So here we will focus on mouse brain. So brain consists of large anatomical structures like olfactory bulbs on the left and like cortex. And this is particular structure, hippocampal formation, which is responsible for episodic memories. And now we are focusing on dentine gyrus. And dentine gyrus is unique in a mammalian brain because it's one of two parts of the brain where new neurons are born, and this, it's hypothesized that this part of the brain is related to the context differentiation for the episodic memory. And so you see this nice animation. We have about 200 millions of neurons in the mouse brain, and now we are focusing on granular layer of dental gyrus. And you see these flashes of electrical activity here. This is how neurons send signals to each other. So here you can see one neuron. On the left-hand side, this dendritic tree receives neurotransmitter signals from other neurons. Then round body in the middle, it makes decision if the neuron should send signal or not. And then on the right-hand side, we have an axon, which is like a wire for targeted dispersal of neuromediator. So now let's take a look how learning proceeds in the brain. So we have these green axons going from entorhinal cortex, and we have this pink dendritic tree of our receiving neuron. And you see these flashes of green axon. This is action potentials. This is actions of neurons sending neurotransmitters towards our pink neuron. And you see that dendritic tree, it has some moving parts which can touch membrane of our neuron, this axon. And then to make process of neurotransmission better, more robust, these neurons form synapse between each other. And synapse is a contact between membranes of two neurons where neurotransmitters inserted, receptors inserted. And receptors are molecules which are sensitive to the incoming neurotransmitter. And then all these 200 million neurons working in coherence to generate behavior of an animal. Okay, now it's time for demonstrations, because without demonstrations, it's not a good science show in the Royal Institution. And here, what we have here today, for your pleasure, is a hydrodynamic model of neuron. You can see here 3 synapses with already with some neurotransmitter poured inside them. So now we will try to simulate with this hydrodynamic model process of learning and generating action potential. So we are starting. So these valves are weights of our neural networks. It's the amount of receptors we have. So if valves are closed, which means that no contact is formed between this cell and this cell, this cell and this cell, this cell and this cell. So we are starting to learn. We are starting to increase our weights. You see, yeah, probably not enough yet. More. You see nice sound of electrical dischargers? So now this neural model is called integrate and fire. And now we see process of integration. Be patient. I hope that we have enough neurotransmitter to fire. Whoa! Integrate and fire. Okay. Now we can do very simple mathematical model, like high school model of neuron. Here we have, on the left-hand side, our red synapses. For every synapse, we have some value x of neurotransmitter released towards this synapse. And then, as you remember, we need receptors to fill this neurotransmitter. So we encode amount of receptors as a W, W1, so every synapse has its own number of receptors. And then we start to calculate effect of incoming neurotransmitter to our neuron. So we multiply X by W to get the overall impact. And then we accumulate all this, accumulated from all synapses. And then our neuron should make decision if it sends signal further or not. And here we are. We need something like a decision function. And this is sigmoid, the most popular decision function in neural networks back then, probably not today. And it gives us nice threshold behavior where we have no activation, where we have low potential. And then if our potential crosses some threshold, we send signal to downstream neurons. That's it. And then we connect millions of these neurons. And the task is to find W's, to find parameters of connections to solve the task. This is what machine learning and AI doing in its way, in our brain doing its maybe in another way. Because we are sure how our neural networks, artificial neural networks doing this, but not sure how our brain is doing this. Okay, now let's take a couple of examples how AI can understand biology. First, let's zoom at the organ level. So what we have usually at many checkups in hospital, we can measure heart activity. So you place those electrodes and then you record for 10 seconds. And you have electrocardiogram or ECG for your heart activity. And this ECG is placed inside your health record. But of course, in your health record, you have a lot of other interesting information, like your diseases, your other conditions, and stuff like that. So scientists from Imperial College London, they took a database of about 1 million ECG records and trained a neural network inspired by computer vision, which they called ARI, and then started to use this model to predict different diseases from the health records. And of course, if you have some cardio diseases, then you will have a pretty good predictive power. But that's not the end of the story. They decided to predict other diseases as well. For example, they took a diabetes 2, which is metabolic disease. So it's like dysfunction of metabolic system, which is not directly related to the cardio system. And amazingly, they were able to predict with about 70% accuracy high risk of developing diabetes 2 years ahead. You see that it's like you are going to hospital. Your current blood levels, sugar level in blood, okay, but you are doing ECG, and then you can be forecast with a high risk of diabetes 2. So you still have many years to change your lifestyle, to change your diet, not to suffer from the consequences of diabetes 2. Because diabetes 2 is really serious disease. It affects 500 million people globally. And it has a very complicated consequences. And I was amazed how AI can find these connections between seemingly disconnected systems, organ systems of our body. And now let's go to the highest, to the most detailed level of our body, to molecules. Of course, you know that we all build of molecules, and in every cell, we have something like a 3D printer, which prints these proteins, building blocks of our body. And sequence, so these proteins, they can be represented in two forms. One form is a sequence of amino acids, which constitutes this protein, and another form is a 3D shape of the protein. And of course, to understand biological function, we need 3D shape, because biological function is determined by interaction between proteins. So to understand this interaction, we need to know 3D shapes, because molecules interact in space, and we need to know how they dock each other. But it seems to be a hard problem. And for 50 years, scientists around the globe worked hard to purify proteins, to crystallize them. And the put them into the X-ray crystallography, which was, by the way, invented here in Royal Institution by Bragg, or cryo-electron microscopy to devise proteins, the 3D structure. And it takes months, and it costs hundreds, £1,000. So then, a few years ago, we started to see all these wonders of deep learning. Many academic and industrial labs started race to use about 100,000 3D structures already predicted from experiment to train their models to predict 3D structure on the same level of precision as experiment. And here, DeepMind was the first again. They've trained their own AlphaFold model to predict 3D structure from the amino acid sequence. And quickly how this model works. So it takes amino acid sequence, then it searches in two databases something which is similar to this protein. First of all, it looks for proteins in other species which is similar to each other. So it creates so-called species alignment. And on the other hand, it searches for database for contact maps, which is some representation of the 3D structure for the similar proteins, for the similar sequences. And then it selects some similar sequences, amino acid sequences, and some similar 3D structures, and then starts to refine them to get final prediction. And by training on these 100,000 examples, they were able to devise a system which can iteratively refine over a few times 3D structure and get quality which is indistinguishable from experiment. And you see, this is very important model. Why? Because before that model, we have 100,000 known 3D structures. But we also knew about 200 million protein sequences without any 3D structure. But with these models, in a days, DeepMind was able to generate 3D structures for all known proteins. So now, you see that anyone who is doing molecular biology with proteins should be happy. Because if you have hypothesis in the evening, then next morning you can query database and get 3D structures you need to test your hypothesis. And that's amazing. You should not wait for many months. And of course, this achievement was distinguished by Nobel Prize in chemistry last year. But I think that, as we've seen, there are many levels. Like we have organ systems levels, organs levels, cellular levels, molecular levels. But I think that the most important level is level of genome, because actually, in some sense, genome connects all these levels together. So let's have some recap what genome is. So of course you know that in every cell of your body you have nucleus. In every nucleus you have 2 meters long molecule, a bit longer than my hands. And this is DNA molecule shown here. It's small, just a tiny part of it. And it has particular structured form. It consists of 4 repeating parts. And these parts, they create a foundation for genetic code. So we have 4 nucleotides, 4 repeating parts, and these nucleotides, or we can call them base pairs. And on the other hand, we have 64 codons. It's 3 letters, like sequences of 3 letters combined, which codes for amino acids, or like stop codons, for example, which regulates translation of the RNA from the genome. And on the next level, we have processes of gene expression. So gene, you can see, if you imagine that our whole genome is like a computer program which codes for our body, then gene is some set of instructions inside this program which has some biological meaning. And here, during gene expression, we have two products for this biological function. One is proteins, and this is called actually gene expression. And the other thing which can be encoded in the gene is non-coding RNAs, and they are required for gene regulation, for regulation of this gene expression. Okay, and as I've said, all our chromosomes, they are like a hard drive or memory for the program which specifies, not fully, but pretty specifically our body. And these monozygous twins, they are proof that this specification is pretty precise. So these guys looking pretty like each other and have a similar body and probably similar physiology or even character. So we have a living proofs among us that inside every cell of our body we have a program which builds our body. So now the task is simple. Let's just decode this program, just learn this language of life. But before we start, we should understand, do we have enough data to understand this, to learn this? And here are data on how much data required to train large language models. These are blue dots. You see that during last years, we see rapid growth of data used to train models. And today, unfortunately, we already reached the saturation point. So we gathered almost all textual information available. So now we need to be inventive to make these large language models better. Because you see, this is a direct limitation of our neural scaling laws. You remember that more data, more compute, more parameters, better we are. So here, we will not have more data in future years. But we will have more data in DNA sequences. Because red dots, it's amount of DNA data, DNA sequences publicly available online. And you see that today, it's almost the same size than textual data. And probably in 2-3 years, we will get the same size, and 2-3 years more, we will have 5 times more than current textual data. Because you see this logarithmic plot here, and you see this red arrow. It says that every year and a half, our genomic sequence data doubles. So you see, 3 years, 4 times more data. Okay. Gena the crocodile. Anyone knows? Yeah, we have pretty many people here knowing Gena the crocodile. So Gena the crocodile is a very kind character from stop-motion animations from Soviet time. And why I'm showing Gena here? Because a few years ago, then I started to build a new AI research institute. I started to think, what is the next big thing? You remember 10 years ago, I thought that after Atari games, the next big thing should be natural language processing and conversational AI. And a few years ago, I also returned to this question. What should be the next big thing? And you know, Gena the crocodile, it sounds similar to genes. So this is why we decided to call our DNA language model GINA, or GINA-LM. So I decided that the next big thing is about understanding language of life, learning it from genomes and predicting all kinds of biological phenomena from it. All what we will be able to predict, we should do this. So with my colleagues, with bioinformatic backgrounds, with natural language processing background, we started this project, and we applied the same methods which were used in natural language processing to train large language models, which is called transfer learning. So in transfer learning, you have two phases to train your model. The first phase is called pre-training, where you just take, in natural language, you just take texts, and then you predict next word. And that's it. You don't need any other data. You just pre-train on this. And then the second stage is fine-tuning. And here, potentially, you can use your pre-trained foundational language model. So you have, after pre-training, you have a model which understands language very well. But now you want to apply this model to some specific task. In natural language processing, this might be like sentiment classification. You want to know if some responses in your charts are negative or positive. But in biology, it can be like these non-coding variants, interpretation, or genome-based disease risk assessment, or genome annotation. So you take pre-trained model, then you gather domain-specific data, and you fine-tune the model to use understanding of DNA language to solve your problem. So this was our approach. And here, how we have applied it to predict promoters for Drosophila. So you see here on the left-hand side, it's part of the gene of Drosophila gene or some other gene. And before the gene starts, you have this yellow part, which is called promoter. And this part is crucial for regulating gene expression. So we want to know where these promoters are located. So what we can do then, we can take the model pre-trained on Drosophila, and then take data which says that this part of DNA is Drosophila promoter, and this part of the DNA is not Drosophila promoter. So you have some set of training samples, and they have a length of 300 base pairs. And then you train your model to solve this task, and you can achieve pretty good quality. So then the question is, how universal our foundational model, how good it at understanding basic rules of genomes. How we can test it? We can test if it will generalize to other species. So what we can do, now we take model, pre-train it from scratch on multiple species, but not Drosophila, and then fine-tune it on Drosophila data. And you see that even if during pre-training our foundational model have seen no Drosophila data, it's still able to solve the task. So we decided to ask if it's sufficient to train model only on human data, no other species. And we did this. So then we pre-trained foundational model, like base model, on human data, and then fine-tuned it on the Drosophila data. And you see, it worked pretty well as well. But of course, not as good as zoo model or Drosophila model. What is another important factor for training these models, for understanding how they work? Another important factor is how much information you give the model to solve the task. So here we took 3 different species and fine-tuned them to solve the same promoter prediction task. And you see these yellow bars. They are for sequence length of 300 nucleotides, and orange bars for sequence length of 2,000 nucleotides. So it's almost 7 times longer sequences. And you see that by providing the same, exactly the same samples, it's the same promoters, but inside larger context, you can achieve much better results. So, of course, now things seems to be simple. Like, we have shown that DNA language models can learn something about genes. We show that it works with downstream tasks. So what should we do next? You are right. We should scale our models. We should make them bigger. And so this is results of one of attempts to scale these models. So here it's just a graph for some specific task. And these bars, these are different models, different genomic models. And this specific 2 bars on the left, they are for EVA model with 7 billion parameters and for EVA model with 40 billion parameters. And you see, unfortunately, that they have the same performance on the downstream task. So we trained model of much larger sizes, and some other models here with the bars with the same performance even smaller. And if you open this paper by Arc Institute, you will see dozens of charts like these, bar plots like this, where these 2 models perform similar on all these downstream tasks. And on one hand, it's upsetting. So what is happening here? Why we have no scaling laws? But on the other hand, it's very positive news. Now we can do something more interesting than just copying what we did for natural language. And here is my hypothesis why just raw scaling is not enough. So if we compare genomes or like genetic texts with the natural texts, we will see that actually natural texts, they are mainly has one scale of interaction, which is like local interactions, which quickly became weak when we move, like when we increase distance between words. But it's not for genes, for genomes. For genomes, probably we have at least 2 scales of interaction. The first one is short-range interactions inside gene of order thousands, tens of thousands base pairs, and other interactions are between genes, long-range interactions on the scale of hundreds of thousands or millions of base pairs. So my hypothesis that current language models, DNA language models, they learn only short-range interactions, and they have enough size to learn everything about specific genes, but they are unable to learn long-range interactions. Could we solve this problem? It seems that it's hard. So imagine that all these dots at the bottom, you see these gray dots. It's like chapters in our books. So it's like, imagine that it's whole Encyclopedia Britannica there, and you have like different articles. And today's large language models, they can effectively read only probable 1, 2, 3, 10 articles, no more. And then you continue reading with the current models, and you go to the next articles. They just forgot everything about what they've seen before. So they have no memory. They can only understand what they see as their input. So what we did, we decided, oh, we need to solve this problem by adding memory. You see, to like read step by step, you need to remember what was before. It's such a simple idea. So we just added memory to that like common transformer, like neural network behind LLMs is called transformer. It's neural architecture, it's the name of neural architecture. So we added memory to it. So during reading some current article, some current input of the model, it can generate representation in memory which is relevant to the task the model is solving right now. And so we can introduce something like a memory highway, where we then take this memory and then use this memory for better understanding of the next segment of our text. So here we just copied, we copy our memory from output to the input, And then we add something, if we need, from our segment 2 to this memory, and then we continue this process. So like accumulating in memory something relevant for our task. So you see, this is probably a nice idea, but will it work? So we needed to test it. And this also became a problem for us, because when we started to look for data sets, for benchmarks to test our model, we've seen that many of them just tens of thousands of words, no more. So we decided that we need to invent something new, some new way to understand what is the longest context to be processed by one neural network. And we decided to use an approach which can be called needle in a haystack. So imagine that this long line of books is a whole Encyclopedia Britannica. And you have 3 basic facts, like Mary went to the bathroom, John moved to the hallway, and Mary traveled to the office. So I take these 3 facts, randomly select somewhere between sentences, and insert these facts there. And I can now control how much context I have and test if for 10,000 words my model can solve this task, or for 100,000 words, or for 1 million words, or for 10 million words. Okay, so let's take a look on results. So this is Google Gemini Pro. So it can solve this task for 100,000 tokens. So you see on the scale of whole Encyclopedia Britannica is this tiny bar there. So what can we do with our recurrent memory transformer? We can do 11 million tokens. So we can find facts and answer questions for 11 million tokens, which is almost 100 times more. So this was published 2 years ago, and it was a world record for the longest sequence ever processed by any neural network. But you see, if you start breaking records, why not to try further? So we have published next architecture in 6 months, which is called associative recurrent memory transformer. And this thing was able to answer where is Mary for facts hidden in 50 million words. Yeah, you see, it's amazing. We like records. So can we apply this to GNLM? So of course we can combine our pre-trained GNLM with this memory. And then we have tested it on the task of species classification. And if you take only 1,000 base pairs and try to classify species, then you have errors about 40%. So now these orange bars is errors. So its error is large. But then if you take 50,000 base pairs, then you can train your model to differentiate species much better. You can reduce your error 100 times. And when you go from 50,000 to 200,000, you can have a gain of 2 times smaller error. So nice. We have a nice technology of memory transformers. We can apply it to DNA. So are we on the brink of solving all the problems? Yeah, here is our nice associative recurrent memory transformers, 50 million tokens. And now let's apply it to full genome modeling sequence. And yeah, the human genome is a 3 billion base pairs. Even if you count 10 base pairs as a one word, it's still 300 million words. It's 6 times more than we had for our task. And you need to train all species available. Is it depressing? No, because now we have more records to break, you see. 3 billion. And today we are looking for resources to gather data and to start training this. Okay. So when we will get this model in a couple of years, what we will be able to do with this? I imagine that as with a understanding language, we will be able, first of all, to predict a lot of biological processes on different levels, on cell type, on cell level. For example, if we provide our DNA sequence like combined model, encoder, like this big model for genomic sequence plus description, natural language description of cell types, we will be able to train model to predict gene expression. If we will combine our genomic model, GeneLM, with some information about like patient condition, we will be able to predict some physiological signals, or like we combine GINA with the ARI and we will get better prediction of diabetes or something like this. Of course, why not? But then you can ask, this is all about predicting something. This is your stupid neural networks, they are just black boxes. How you want to make something, how you can extract something meaningful out of them. Because we want not only to predict something, we want to understand basic principles behind our cells, behind our biology. And here, AI has something to apply to. And this is called feature attribution. So for example, let's take a look how we can measure how different parts of our input sequence affect our prediction of end of the sequence. So we have Heathrow Airport is located in the city of, and large language model should predict the next word, which is London. And with the feature attribution methods, what we can do, we can calculate from the pre-trained model which words are most important for doing prediction. So here, of course, you can see that Heathrow is the most important word, and airport and city are the second most important words. And there are some other words, they are not so much important. And of course, if we trained model for some biological function, then we can try to do the same trick with the input attribution. And we did it. It's from our paper published this January in Nucleic Acid Research, where we have take our genome model, pre-trained genome model, and then fine-tune it on the task for predicting sites, like along DNA, which parts of the DNA are binded by some transcription factor, which is called CTCF. And you can see that after model was trained to predict binding, then with feature attribution, we were able to find parts of the DNA sequences with the highest importance for this task, and motifs underlying these patterns of DNA letters, of nucleotides underlying these parts of the DNA, they confirmed by other studies that these are really the patterns where CTCF is binding. What we will be able to do potentially with this? You see, after we have trained the models on all these different levels, then we can go back and find parts of the DNA associated with all these processes. And you see, this can provide us common ground to connect somewhat disconnected processes. For example, here, if you have on top parts of the DNA which are related to some cellular processes, and another part of like important sequences which are related to some organ, and then you have some intersection of these instructions, important instructions, then you can say that if these particular parts links this cellular level with the organ level. And on the other hand, you can say that something which goes beyond in regulation of genes on the organ level, which is not important for cellular level, this is what makes this organ like functional, what orchestrates many cells to work together. And of course, we can do this for the whole body level too. Yeah, and how we can apply this to, just to give you an example of some fundamental biological problem. And this is actually the problem we are actively working on in our London Institute of Mathematical Sciences from the theoretical side, but we can support our theories, because today we have like two competing theories of aging. One says that aging is accumulation of errors, and another says that it's pre-programmed. So how we can use DNA language models to differentiate between these hypotheses? I think that for the accumulation of errors hypothesis, we should have something like if we collect the data for different species, with the different lifespans. So you see, like we will take mammals with the different lifespans, and then we will fine-tune our model to predict lifespan from DNA of this species. And after that, we can go back and reverse engineer, find input sequences relevant for this prediction. And I think that if the theory of accumulation of errors is correct, we should see something like a blurred signal, because all the parts of our body contributes uniformly to our aging, like they participate uniformly. But if other theory is correct, that we have some pre-programming to turn on sentience switch, then we should have some specific parts of the DNA which are very important to predict age of the animal. So this is how we can use these models. And now, at the end of my talk, let's do some leap into the future, what it might look like a few years ahead. So imagine that I took GeneLM and asked, hey, GeneAI, can you create me a lion that's the size of a domestic cat, like a pocket lion? And I have all this generation, blah, blah, blah. Looks familiar, yeah? Now you have a genome sequence. Would you like me to send this code to the nearest Genforma lab? I don't know what Genforma lab will be for incubation and growth. Yes, please send it there. Okay, 90 days from now. Your imagination already drawing pictures, yeah? What will happen 3 months? Yeah, of course. A cute line. So I think this is what future holds for us in AI and biology. So and tonight, I think that we have pretty confidently charted clear roadmap towards it. So what is our roadmap to decoding life with AI? So first of all, we start with the understanding that AI can learn the genetic code, the programming language of life, through genome-scale DNA language models. And then these models will enable us to predict and interpret molecular, cellular, and organ-level phenomena just from genome sequence. And as more and more genomes are sequenced, we will decipher, process across all known organisms. And this will empower us to answer biology's deepest, most fundamental questions and improve human health. And this is what we are working on at LIMS, actually. And at the end of this talk, I would like to thank all my colleagues at LIMS for great intellectual environment they create, where you can pursue crazy ideas and do really important stuff. And thank you for your attention tonight.